I'll start by saying that what I'm going to talk about in the next 40, 50 minutes or so is about a work that is published. The name of the work was Assessment of Network Modular Identification Across Complex Diseases. And it's a collaborative effort. It was really spearheaded by a senior postdoc in my group, Daniel Marbach, here. And that's why I thought it was really fair to give him the last authorship or last co-authorship position on this paper. And a lot of work was done by Sabanaz, who also at the time was a postdoc in my group. And then we have other members that made significant contributions, either by being like Julio involved in the whole Dream Challenge system of how to set up these competitions, these collaborative competitions. And also we had several authors from the winning team. I'm going to tell you more about what actually was a competition, but at the end of the day, it was quite a lot of people who were involved either by participating and also by really doing the analysis of the results. So that's why I thought I'd give you a little bit the faces and the names at the very start. So the basic biological background to it is about networks, and you are assumedly familiar, maybe even have worked yourself on biological networks. There are many different kinds. So there could be protein interaction networks, metabolic networks, co-expression networks, homology networks, signaling networks, neuronal networks, networks between species, even networks between diseases, as you will see today. A lot of times it seems to make sense to try to present our knowledge in terms of nodes that are particular elements and their relationships that are then represented by the edges between these nodes. And the information contained or gleaned from biological systems is then represented by such networks. And biological networks many times are very modular. And that means that subnetworks are much more densely connected than you would expect by chance. So these dense subnetworks often have also been called network modules. And identifying them from the network is quite a challenging and important task to be done on these networks. And many methods have been devised to do this, and they take quite different approaches. I'll just go over kind of the most popular methods to try to do this, to extract a densely connected subnetwork from a large network. One kind of method is to do kernel clustering. That is the network, for example, where all the nodes are genes, is translated into a similarity matrix or adjacency matrix that kind of defines the networks, where every pair of two nodes can either be connected or not, or it even can have a weight that kind of determines the degree of connectedness. And once you formulate this in terms of a similarity matrix, you can then basically use tricks in linear algebra to transform this matrix and to cluster it. And many methods have been devised. It's basically then a linear algebra problem. You could, for example, diagonalize this matrix and look into independent components, and a whole bunch of matrix methods have been developed. Quite a different approach is to say, okay, what actually is modularity, and give it a definition and a score. And then you would say, okay, let's seek some global modularity measure that is independency of how these subnetworks are defined. And then I basically turned the whole question into an optimization problem. And then you have a whole army of algorithms that have been developed for optimization, and you basically try to seek optimal solutions in this global space of where densely connected networks could be. So that's modularity optimization. Another approach is again completely complementary. It's based on random walk, where you present your network and let a virtual entity walk along the network, and allowing it basically only to have these walks across or between nodes which are connected. So it will then walk on the edges, and you just observe that these virtual entities will spend more time on densely connected sub graphs. And that's again by just then looking where these virtual entities have been, you can detect the densely connected parts of a network. And another method or collection of methods is to start with local seeds and basically just explore the neighborhood in an agglomerative process that would basically collect edges that are connected to a particular seed, and then kind of grow them, but will not be able or will stop to grow when there is not a lot of connections into other parts of the network. And that's kind of also a very well-known method to try to identify well-connected or densely connected subnetworks. So these are just 4 typical methods, and other methods, which I refer to as ensemble methods, have basically tried to see, okay, these other methods, they use different properties to find densely connected subnetworks. Maybe by combining them, we can do even better. So these are ensemble methods, which try to then only define modules if more than one method is kind of voting in favor of such a module. And they either merge or overlap the predictions from 2 or more methods. Okay, so we have many methods to look for network modules, and the question is, which one do actually work best? And the important word here is best. So that, of course, implies that there is some scoring involved, and the other question is, besides what do you score, is also what do you score? So which kind of networks, for which kind of networks do we try to identify modules? And in our explicit case, we obviously looked at gene-gene networks, but we tried different types of networks, and kind of also asked the question a little bit more globally: are there general methods or types of methods that always or most of the time outperform other methods? And then in terms of how we scored, this is what most of the talk will be about, that we came up with a new scoring method, and it was a new way of doing it that used completely complementary type of data to do the scoring. So that was kind of the novelty in our approach. Now, what I want to kind of highlight, and for good reason, is that the whole approach of this Dream Challenge is to decouple the method development from the method evaluation. So that, I think, is very important for reproducible science, because in bioinformatics, more often than not, people who invent methods are also the first to develop them. It's almost that the quality of a method is given into the same hands as the developers, and that introduces, of course, a massive bias, because it implies that imagine we would do this everywhere, that the people who now designed the new vaccines against COVID, they would also be completely in charge of assessing the validity of the medicine. And of course, you cannot do this. You have to separate this. And it's not frequently done, interestingly, in computational biology. A lot of publications come out with a new method, which then is compared against the competitors. But the people who propose the method are the same people who are doing the comparison. And I think in general, this is not really great. So we need competitions that where everybody has kind of the same, you know, difficulties to prove the validity of their method. I mean, maybe some of you have heard now about the challenge, the CASP challenge of predicting protein structure from just from the nucleotide sequence, so to speak. And also this is an excellent example of that by decoupling, by making the evaluation, decoupling it from the people who develop the methods, you know, you have an unbiased evaluation. And that's of course also a perfect setting, as it turns out, then to develop machine learning methods. And I don't know if you heard about the details, but I think there is a revolution has been going on there. I will tell you about a different challenge, the disease module identification dream challenge that I helped organizing. And I want to point out at the start that the person who was really spearheading this project is Daniel Marbach. He was a postdoc in my group at the time. And he has actually moved on now to industry. And he deserves, I would say, most of the credit for the project. So what have we done? I'll give you, since we have a little bit of time here, a background that will motivate how we set up the strategy to evaluate the modules. And for this, I have to go back a little bit about how genome-wide association studies work. And maybe I can just ask you at this stage, who is actually familiar with genome-wide association studies? Can you just raise your hand in the panel so that I can see it? Okay. Wow. I don't see, it doesn't seem to me that you're all GWAS experts, so I'll tell you a little bit about the details. So what you see here in this slide is an example of the genetic variation in terms of single nucleotide polymorphisms across, you know, just an example of 5 individuals. They happen to be members of or participating in this challenge. So you may see these faces in a different context again. We have 2 chromosomes, one from our mom, one from our dad. And every here and there is a position where there is variability between humans. And that is the genetic variation that to some degree is responsible for phenotypic differences between humans. And a typical example is to look at a complex trait, like, for example, human height, and then ask the question to what extent the variability in this trait can be explained by any such single nucleotide polymorphisms. And that is what is called an association with the genotype. Now, as we have 2 alleles, one from our mom and one from our dad, there are 3 genotypes. And in the most simple case, well, if you want to ask the question whether a phenotype has been affected by a genotype, it can be posed as a linear regression question, where equation, where you just express the phenotype as a linear function of the genotype and have some effect beta. And the most standard way of coding your genotype is just to count the dosage of the minor allele. That would be the G in this particular case. This is, of course, a particular coding. It doesn't have to be like this, but it's kind of the most simple thing one can do. Obviously, it's kind of between the recessive and dominant inheritance mode. Now, the important point is that this is then an really statistical problem. And what you do with this data is to estimate your effect. That would be corresponding to fitting a line through this data of the 3 coded genotypes, but also to estimate, of course, the error. In this particular case, as you've seen, the distributions are quite wide, and the effect, even though it seems to be some sort of effect, is not large enough to also be estimated to be different from 0 within the error margin. So what you really need here as a most important variables is the effect estimate, but also the estimated error. And that's what then what you can use to assign a normalized effect. Now, that is very easy for doing this for one particular genotype. It gets a little more complicated due to the fact that we have so many different SNPs of the order of 1 million or so in human populations. And in the most simple case, it really boils down to a screen where every of these genotypes are then associated with a particular trait. And the significance often is kind of plotted as a function of the chromosome position. These are so-called Manhattan plots, in order to fish out the very few super significant associations where the effect sizes within the estimated errors are large enough to be deemed so significant. The main challenge in this approach is because you're making so many tests that the significance has to be really large because you have to correct for multiple hypothesis testing. This approach has been performed now for over a decade, and by these genome-wide association studies, almost every position in across the various chromosomes has been associated with one trait or another, whether it's different diseases, disease risks, or even just any kind of human traits. So a lot of loci have, through this genome-wide association studies, have been, if you like, annotated of being implicated in one or several human traits. Now, for us, if we want to use this information to say anything about genes, we have to think how to map the significance values that are computed at the level of SNPs, so that's ultimately nucleotides, to genes. In the past, the simplest approach was simply to say, okay, I have a position on the chromosome that is very significantly associated with the trait. I will just find the closest genes and kind of by implication say that probably this gene is then associated with this trait. I think one can do better and do better statistics, and methods have been developed to do exactly this. So in the kind of simplest version, what you would do if you want to map the SNPs to a gene is to take a window around the gene, usually of the order of, I would say, at least 100 kilobases from the transcription start site, upstream, and from transcription end site downstream, have quite a big window, and then to see if there is an aggregate effect across the SNPs, because of course they cannot potentially just have an effect on the protein, so literally altering an amino acid, but also, for example, be slightly altered binding sites, have to do something with a chromatin conformation. There are many ways of how nucleotide variants could impact the gene or its expression. Now, the thing is that to do this aggregation well and statistically sound, one has to take into consideration that the genotypes themselves are not independent. There is something which is called linkage disequilibrium, which implies that there is a trend that when two SNPs are proximal on the chromosome, that there is a residual correlation between them. So they're not completely independent, and that is represented here by the SNP by SNP correlation matrix. And this then is also transmitted actually to the effect sizes that you estimate on this by genetic associations, which means that, for example, if you have several strong associations proximal to each other, it can then either happen because both of these SNPs are independently associated with the traits, or these SNPs are already very similar to each other. That means if you have the SNP profile of in the population of one, you could easily predict the other one. So most individuals, given the one SNP, you will know what's the value of the genotype of the other one, and then this kind of very similar significance is just a result of the genetic correlation. And if you think of it, if you want to vote whether this gene is important or not, it makes a big difference how we count contributions that come from correlated SNPs and those that come from independent SNPs. And I will try to explain now how you do this statistically. So imagine we just look at two SNPs and we look at the distribution of the effects, and we see actually that these effects are correlated with each other. Now you want to ask the question, if you make a sum of chi-squares, so you assume that they are normal under the null, but that you sum up the squares, and you get this sum statistic, which I call A. And simply the question is, how large a value of A do you actually deem to be, let's say, not inconsistent with the null? So very, very, very large. And now this depends on the correlation between the two SNPs. If they're completely independent, you have a sum of chi-squares of 2 degrees of freedom, and you know actually how to do the statistics. However, if they're not independent, then that's really different. And what you would then have to do, so you cannot use this assumption of independence, and you would actually have to do very simple mathematics to rotate into a new basis where the two SNPs are independent, as shown here. So this is just a rotation from Z1, Z2 into Z1 prime and Z2 prime. Here, the two axes of variation are independent, and now you can just work under the assumption that these contributions are independent. However, the rotation, as you can see, now means that the magnitude for the two variables is not the same. Basically, the way it's done is that you look at the correlation matrix and find the new eigenvectors of it, and this is your new basis. But this will then imply that you will have the eigenvalues as weights in front of these chi-squares. Now, it turns out that a weighted sum of chi-squared variables, you can still relatively easy actually determine the null distribution. It's you don't find it in a textbook, or MATLAB doesn't have it as ready functions, but there are tools that can kind of semi-analytically compute these distributions, so that you have a basis to do this basically analytically. And that's exactly what we did in order to find the gene scores. And the tool that we developed is called Pascal, for pathway scoring algorithm. And it's basically the main improvement over an existing tool named GWAS, was that this tool used, in order to compute the null, a permutation approach. So that had limitations. First of all, it was much slower to do it this way. So you see that the runtime of GWAS here was, I would say almost 2 orders of magnitude higher than if we do it analytically here with Pascal. These are the orange markers here. And on top of this, of course, if you do it by permutation, the precision is limited, because the kind of smallest p-value estimate that you can give is 1 over the number of permutations. So in Pascal, they choose to do this at the order, I think they made 1 million or so, or maybe 10 million reshufflings. And that's basically where the cutoff for the p-value was. While if you do it in Pascal, you can see that the degree of precision of the p-value is much higher, because you have a basically analytic approach to do this. So this Pascal tool really changed the way things could be done in computing gene scores. And the basic idea is that you have a reference population, for example, the HuBMAP panel or 1000 genome panel, and you use the SNP correlation matrix computed once and for all the neighborhoods within the gene, and then have the gene scoring. You can set up it quite quickly, and then basically for any GWAS that comes in with the summary statistics, very quickly compute gene scores and get a p-value which is gene specific. And so that was done both to do the scoring of individual genes, but also for pathways. And in pathways, usually you want to come up for a significant score of summary statistics that have contributions then of all the genes that are associated with the pathway. A pathway could be a Go annotation, a KEC pathway, It could be the targets over transcription factors, any kind of set of genes annotated to participate in a particular function. And there again, we have the subtlety that sometimes there are genes, you see this here, two genes that are proximal to each other, and the contributions that it would get at the SNP level are actually overlapping in the SNP-SNP correlation matrix. So what we came up with is a method that would use, if you see something like this, that two genes are coupled, you would define a so-called meta gene. So pathways would then, if they include two or several genes that are actually in linkage disequilibrium, consider them as a joint entity. And that, again, is very important in order to reduce the number of false positives and doing the statistics right. Okay, so that's a little bit of the methodology. I just give you an example that if you don't do this fusion, you have massively inflated p-values on a null data where there's actually no signal just because you didn't do the statistics right. And if you do this fusion, basically things come out pretty well calibrated. So that was kind of a little bit introduction to GWAS and the gene scoring methods we developed. And now we actually use it for this challenge. So I'm coming back to our networks and how to get densely connected subnetworks or modules out of these networks. And the approach is actually quite simple. So in order to define or to answer the question whether the participants of our challenge managed to get good modules, we used our GWAS gene scoring and pathway scoring procedure. And I'll explain to you now how we do this. So the idea of the Dream Challenge is to really foster a community effort to systematically assess the module identification methods of a diverse set of genomic networks, and as a kind of secondary goal, also discover novel pathways underlying complex disease. And the timeline was as follows. So this project actually started more than 4 years ago. And we had the challenge in at the end of summer to October 2016, where the participants would, you know, contribute their modules that they concluded from or generated from these networks. There was a conference quite close after, and then actually took more than a year to process all this data and come up with the paper. We had actually more than 400 registered participants. I was asked how many people were there involved. So more than 400 of external participants. And there was a lot of posting and discussions going on in the forum, which is part of the architecture of this Dream Challenge. Now, just as a little bit of a background. So when you do a pathway analysis, often what you do is, first of all, you take a pathway from a database, GoCAC, Reactome, an annotated set of genes. And then to test whether this pathway is enriched in the data that was then usually generated on a relatively small scale for a particular type of experimental setting. So for example, take a cancer and compare the gene expression as compared to some control. And then the idea is simply, is this omics data, if you project it on your pathway databases, enriched in any of these pathways? And this would give you the enriched pathways. Gene set enrichment analysis is kind of the classical tool to do it. There is a different type when you do a network analysis where the omics data is not analyzed directly, but is first projected into a network. And that works in particular if you have large-scale omics data, where you can, for example, just use the similarity matrix between the genes across this omics data, if you think of co-expression. There is, of course, also some omics data that directly gives rise to networks. For example, a protein-protein interaction could directly be seen as a kind of network property, where you simply ask, are the proteins of, coded by these genes, do they physically interact with each other? Now, then the idea is, once you have it in a network, to break these networks down into modules, and then simply test whether these modules are, again, enriched in any of these pathways that you have in your database. Now, our approach was different. So while we kind of started at the network level and then asked the participants to do the module identification from these networks, so using this as a challenge task, we did not use the classical pathway databases, but instead we used GWAS data in order to do the annotation of the modules. And the main motivation was that, well, first of all, that these pathway databases are biased in the sense that they reproduce a lot of simply what has been researched, and there's a lot of potential gene sets that are actually functionally related, but that simply haven't been studied yet in a dedicated way. So we thought that this GWAS approach was quite complementary. Of course, you have a bias also about what human traits have been studied by GWAS, but once you ask a trait, is it associated with genotypes, you get an answer for all the genes. So in this sense, it's kind of not biased in terms of what genes we're looking at. And so we used GWAS to do the annotation, and I will tell you a little bit more. But first, about our networks that we offered to the participants of the challenge. We had 6 different network types. 2 were protein interaction networks. One was from String, that's a Swiss effort, and then was another. They differ a lot by the number of annotated edges they have, but cover a big portion of the human genes. There was a signaling network, a co-expression network. You see it had exactly 1 million edges. That's just a cutoff in terms of the most significantly co-expressed gene pairs. Then there is a cancer dependency network that was used and a homology network. They differ a lot in the degree distribution, obviously in the biology that they present. And we wanted the participants to come up with methods that work well on all these type of networks. Now, so how was the design of the challenge? So the networks were given, but not obviously with the gene annotation, but we replaced gene names by numbers, by random numbers that were of course the same for everybody. So they could not use any external information that might have been available for the genes. But only the network property, so the topology and the strengths of the network edges, to then identify the modules. And these 40 teams that were participating in the main challenge simply had to give us, for each of the 6 networks, their set of modules that they generated from these networks. And then we used Pascal as our Pascal scoring algorithm to ask how many of these networks would turn out to be significantly enriched in genes that had SNP summary statistics that made them significant in our Pascal approach from a large compendium of GWAS. I think we had more than 200 different traits, so it was quite broad across different phenotypes that had been investigated with GWAS. And as long as the module would bring up genes that were supported together to pick up signal in any of this GWAS, we would say, okay, that's a good disease module or human trait module, and well done, you get 1 point for this module. But if it didn't pick up signal from any of the GWAS, you would not get a score. So then we did another challenge, which is slightly different, where the 6 different modules actually could be co-analyzed in the sense that the numbers of the genes would then be the same across the different networks. And we thought that maybe participants could use this to define more robust modules that would appear across several networks, and that there would be very smart methods that kind of could outperform the individual network designs. But that was an open question, whether connecting the different modules, the different networks would actually bring any benefits. Okay, so the submissions had to be done in a way that the modules had to be non-overlapping, so disjoint. That was mainly a design feature, because otherwise we thought it would be very easy to define a core module and just decorate it with other genes. And so, basically artificially increase the number of significant modules. The number of module was not fixed explicitly, but the module size. So it had to have at least 3 genes, but not more than 100. That, of course, again, because your modules could have been at least of 3 genes, meant that the number of modules could not exceed the number of genes divided by 3. But otherwise, there was no constraint. And as I kind of alluded to, simply the score was the number of modules showing enrichment in at least 1 GWAS. And we controlled for the false discovery rate when doing these enrichment scoring. So then the question is, did any of the methods win? The participants had to make available to us the code, so we kind of could look what are the different methods, what approaches do they take, and here are the results. So what is shown here is every number is a method. It's colored according to kind of the method type that I explained before. This year is the overall number of the score. That means identified modules across all networks. For example, this is the best module. It got ranked 2 because we had a community effort that I will explain to you as well. But of the individual methods, that was the best method. And it was followed by several other methods using also kernel approaches. And so what you could see as a very first result is that the best performing method did not all come from the same method type. And that was, I think, very interesting to see that there is not one method that is basically out competing all the other method types. The good methods performed well across all the different types of individual networks, but that could vary. For example, the one which kind of won the challenge on signaling did not do as well on co-expression, so it wasn't in, I guess, in the top 10. And there were other methods that did better, but on the whole, so the overall score, this was the best method. And then, of course, we kind of asked general questions about, you know, what could be learned from the comparing the different methods. So as I mentioned, the top 3 teams came from different categories. And the other thing which is kind of really interesting is that we also did so-called community predictions. So I will try to explain how this worked. It was basically taking all the methods that were better than the median. So I think there was 20 methods or so that were combined. And then there was made a consensus whether they had assigned 2 genes to the same modules. And if that had been, that was kind of the new edge, so to speak, coming out of this method. And then we actually used the top team's method to reanalyze the new similarity matrix, the consensus network, if you like. And that came up with new modules, and that was what we call our community prediction. And it did ever so slightly, I think by maybe 5 modules or so, better than even than the top method. It's a phenomenon that is well known, like crowd wisdom, it's often referred to, that method by aggregating across different methods, you can do really even better than any of the individual methods. However, note that the margin is not extreme. You do a little bit better, but it's actually computation. It's very expensive because you would have to compute over 20 methods and do this. So it's not really practical. It's just to see that you could still push the boundaries of the individual methods. Now, here is a diagram showing the individual similarity of module predictions. So every single element in this graph is showing a result from what method it came from and from which network. So the shape of the marker is which network, and then the color is used for the method type, and then you have individual predictions. And we projected all of the module outcomes into this principal component space. And what you can see is that mostly the variability was driven by the different networks. So of course, if you analyze different data, you also get outputs which are more similar if it's on the same network, that's good. But that within the networks, there is still quite a lot of variability. And the top performing methods are usually actually quite close to each other. I mean, not exactly the same, but here's a winner and the follow-up second place. And the output is quite similar. In general, the overlap between the modules generated by the different methods overlap within the networks. You know, for about 20%, we found that the overlap between the methods is very strong. So these are really consensus methods that would have been found by most of the methods. Sometimes you find things that there is a module that has a submodule by another method that is almost completely contained. So these are strong overlap and submodules, here 17%. And so depending on how much is the overlap, you could see that roughly 50% of the modules are consensus modules. Everybody sees them more or less. But actually more than half are kind of only modules found with weak overlaps to others and with insignificant overlap. If you look across networks, because of course different networks to some extent still capture similar biology, the overlap is even much smaller. Now, we thought it could be an interesting question to ask if the module size, if there was a sweet spot, so to speak. Was it good to make small modules or big modules? As I mentioned, it had to be between 3 and 100 genes. But it turned out that this is actually not the case. Obviously, if you do smaller modules, you can produce more modules. So it's a little bit potentially, the way you hedge your bets is better to get a more significant, but it didn't really work. That, you know, some methods which had a lot of small modules didn't get a lot of really significant modules. So our winner is right in the middle. On average, has maybe 30 genes in the module, but the other well-performing methods could have either significantly less or more genes. So there is clearly, for our challenge, there was not a winning granularity. Another question we asked, whether modularity as a measure, and I'll tell you in a second what that means, is something that if you optimize this, really brings you to the to be the winner. And again, that's not the case. So maybe let me just first say, what is modularity? Modularity is basically within a module, you're asking how many of the potential links are actually realized. It's kind of asking in a friendship network, how many of your friends are also friends amongst each other? Are you fully connected? And that can go up to 1 if it's fully connected, so super dense, and 0 if, you know, none of your friends are friends amongst each other. The modularity scores, average modularity scores about the output of the different methods did not kind of very decisive on whether you could be winner. There are other methods which did not bring up so many disease modules, so those supported by the GWAS, even though the modularity score was very high. So it's not true that just fishing the most dense modules is the best way to win the challenge in terms of disease modules. Okay, so as I mentioned, there was a sub-challenge where the networks had been aligned, and it turned out that none of the teams who took on the sub-challenge number 2 outperformed the other methods. So our consensus prediction, which came up with, you know, 23 modules, was doing better than any of the other methods. But the methods that kind of tried to combine the different data sets were actually not better than those which used just a single network. So here you can see the different methods and which networks they used. So for example, the top one was a kind of a high score of the individual ones, used just the 2 protein-protein interaction networks. But the score was not significantly different from a network that just used one of the networks. So at least there was no dramatic performance improvement. And that's why we said the sub-challenge 2 was left vacant in terms of a winner. There was not a clear demonstration that people could make really use by co-analyzing these networks. All right. So another question we examined retrospectively was whether the modules that were found were kind of particularly enriched in one trait or another. And that turned out not to be the case. The modules that were found really got enrichment in traits across different categories. So you see there are some which have to do with mental health, and in particular schizophrenia, which has been studied a lot by GWAS, was very powerful in declaring modules to be relevant. It really goes across different traits. So these networks did not in any particular way point to genes that are important for one trait or another. And that probably makes sense because these modules, if you look at protein-protein interaction networks, of course, know intrinsically nothing about any particular trait. And they can be, subnetworks could be relevant for any of these traits. So maybe I skip this. What we also try to do is to use the module outputs across the challenge to try to infer a disease network. So every node in this kind of illustration here is a disease or a human trait. And the edges are then in terms of shared modules across the entire challenge. And what emerged is that, of course, many diseases which are kind of known to be related, for example, these are all lipid traits, and they correlate very closely to coronary artery disease. Of course, people know this, that your blood lipids are a major risk factor for coronary heart disease. So having them kind of clustered close to the lipids, that does make a lot of sense. They are other kind of more disjoint groups of diseases, which are known, for example, to have a strong inflammatory component to them. But some connections kind of were maybe not anticipated. You know, if you think about psychiatric diseases, do they more relate to diseases that have an inflammatory component to them, or maybe to more the things in the related to your health in terms of body mass index? Well, they're kind of in between, and I think that just was an interesting visualization of how the diseases would be similar in terms of how modules had been attributed to them. Okay, so finally, you could ask, what is this really all good for? Why do we want to have these modules? And I want to kind of show you one case that can illustrate the potential use of these disease modules. This is a module that actually came out significant for height. So the GWAS of height supported this module, but actually kind of marginally. You see, this is a corrected p-value, is just above below our nominal threshold. And that's mainly because there were 3 genes that the GWAS had attributed a sizable effect to it. And all the other genes came in kind of guilty by association because they were in a densely connected subnetwork. However, this would mean that many of these genes, because they are so close in the, I think, protein-protein interaction to these players implicated by height, could also potentially have a role in height. So this is how you can make predictions about genes that are relevant for this trait. And indeed, if you look at the summary statistics of height that had a much more highly powered studies 4 years later, many of these genes would have in the time then picked up sizable effects. So as illustrated by the color code. So this module, the very same module, was much more significant when evaluated by the height summary statistics of this follow-up study from 2014. Now, and there are actually also other omics data that supported the relevance of many of these genes for height. So, and there are other examples where you can look at the modules and then look at the kind of other support that we get for the modules. and see that often the GWAS implication is indeed also supported by other omics data. I'll not go into the detail, but there is a kind of a transfer of knowledge here that is potentially quite useful just to learn about other genes that could be implicated in a particular trait. So what I also want to mention that learning about the methods was really very important. So for one thing is we had 3 extremely good methods that are very complementary. And Mattia from my group set up a toolbox which has all the 3 methods in one single package, which he called Monet. And he also justified or kind of retrospectively analyzed these methods again and looking at their performance in terms of computation time, RAM usage, and how well they do on in silico data, and simply made the methods available to the community. So if you have a network clustering challenge, I would recommend trying out this tool. So, well, with this, I guess I come to the end of the presentation. My take-home message is that this crowdsourced challenge is really very useful to enable a critical assessment of modular identification methods. We showed that several top approaches recover complemented disease modules across a diverse set of molecular networks. And finally, the molecular network modules, they really reveal core pathways underlying a variety of complex traits and diseases. And as such, I think an asset to try to annotate genes in the context of GWAS and the networks. So with this, I close. I want to show again the various collaborators, particular credit to Daniel Marbach, who led this challenge, and Savanas, who did a lot of work in analyzing the data. And of course, many of the other contributors, both in setting up the Dream Challenge and contributing methods. And I also want to show some members of my group who were involved in this, and Zoltan, who's a professor also in my faculty. And with this, I think I'm going to close and would be really happy to get questions from you.